Welcome to the Duck Dive. Today we're offering you a shortcut, really, into some complex but super important research. We're looking at a molecular showdown, basically a contest to see which lab technique is best at finding those really faint traces of life hidden in the environment, specifically in water. We're diving deep into environmental DNA, eDNA, focusing on this key paper led by Guri out in Ecology and Evolution in 2024. The full title is Quantifying the Detection Sensitivity and Precision of qPCR and ddPCR Mechanisms for eDNA Samples.
Yeah, it's a bit of a mouthful, but the heart of it is a really rigorous comparison. They took the two main ways we quantify DNA, that's quantitative PCR, or qPCR, and droplet digital PCR, ddPCR, and directly tested them against each other.
To see which one is better at finding tiny amounts.
Exactly. Better sensitivity, meaning finding it when it's rare, and better precision, so getting the count right once you find it. They wanted hard numbers on that.
And this isn't just, lab bench stuff. Why does this actually matter out in the real world?
Oh, it's huge, because reliable eDNA results are now fundamental for ecology. Think about tracking an invasive species early on, or checking the population of some really elusive fish.
Your whole conservation strategy might depend on knowing if it's there and how much is there.
Precisely. Management decisions hang on getting that DNA count right. Accuracy is everything.
Okay, let's step back a bit. For anyone maybe less familiar, what exactly is this eDNA we're talking about?
Right, so eDNA is basically the genetic junk that all living things shed. Skin cells, mucus, waste products. It all ends up in the environment.
So a fish swims by, leaves some slime or scales, and we can detect its DNA in the water later.
That's the idea. It's revolutionary because it's non-invasive. You don't have to catch the animal. But the big challenge, especially in, say, the ocean or a big lake, is that the concentration of the specific DNA you're looking for is often incredibly low, vanishingly small amounts.
So you need tools that are exceptionally good at picking up that faint signal. Hence the need for sensitivity and precision.
Exactly. That's where these methods come in.
Okay, let's start with the classic, qPCR. It's been the workhorse for a while, right? How does it actually count the DNA?
Yeah, qPCR is the established one. It works by amplifying the target DNA segment over and over, and it uses a fluorescent dye that glows brighter as more DNA copies are made.
And you watch this glow happen in real time.
Right. The key metric is the cycle threshold, or CE-value. That's the point, the cycle number, where the fluorescent signal crosses a set baseline. A lower CT means more starting DNA, a higher CT means less.
But it's not a direct count, is it?
No, it's indirect. That CT value doesn't tell you X copies. You need to compare it to a standard curve.
Ah, the standard curve. So you run samples with known DNA concentrations alongside your unknowns.
Exactly. You plot their CT values against their known concentrations, draw a line, and then use that line to estimate the concentration in your unknown sample based on its CT value.
Okay, I see the potential issue there. Your final number completely hinges on how good that standard curve is.
Precisely. Little errors in making the standards, pipetting variations, slight differences in reaction efficiency between runs. It all introduces uncertainty. And that gets magnified when you're dealing with low concentration, often noisy environmental samples.
Which brings us to the challenger. ddPCR. How does that try to overcome these limitations?
ddPCR takes a radically different approach. It doesn't rely on the rate of amplification. Instead, it uses partitioning.
Partitioning, you mean dividing the sample up?
Exactly. The machine takes your sample and splits it into thousands, often tens of thousands, of tiny microscopic droplets, each encased in oil.
Wow, so each droplet is like its own tiny independent PCR reaction.
You got it. The PCR happens inside each droplet. Then at the very end of the process, the machine simply counts how many droplets contain the amplified target DNA they glow positive and how many don't they stay negative.
So it's a simple yes-no count for each droplet.
Right. And based on the fraction of positive droplets, using a type of statistics called Poisson statistics, it calculates the absolute starting concentration of DNA.
Absolute, meaning it doesn't need that external standard curve for the basic quantification.
Generally, no. That's the big theoretical advantage. It's like instead of estimating weight by comparing density, you're actually counting individual grains. It promises much higher precision and accuracy, especially when DNA is scarce.
Okay, theory is one thing. Let's get into how Guri's team actually tested this head-to-head. What was the setup?
They were smart about it. They didn't just use clean lab water. They used 153 real seawater samples collected over several years from 2 different fjords up in North Norway.
Balsfjord and Frackfjord, right?
Yep. And they sampled at different depths, surface, middle, and bottom, taking duplicates or triplicates.
Using real-world samples with all their natural complexity is crucial for results that ecologists can trust.
And what were they looking for in that fjord water?
They targeted 3 common and important fish species there: Atlantic cod, Atlantic herring, and saithe.
Okay, so real water, real fish targets. How did they structure the comparison itself?
This was key. They ran the exact same samples through both qPCR and ddPCR systems. That includes the environmental samples and, importantly, a series of lab-made standards with precisely known DNA concentrations.
Covering a huge range, I saw, from almost nothing up to very high concentration.
Yeah, from 3 copies per microliter, incredibly dilute, all the way up to 1 million copies, $167. This allowed them to see how each method performed across the entire spectrum.
And they use a specific statistical method, Bayesian inference. Why go that route? Seems pretty advanced.
It is, but it was necessary for rigor, especially down at those super low concentrations where results get fuzzy. Bayesian methods are really good at handling uncertainty.
So it helps give a more robust estimate of the true quantity and, critically, how confident they can be in that estimate.
Exactly. Let them model the quantities from each method independently and then rigorously compare not just the average result, but the spread of possible results, the precision for both qPCR and ddPCR across that whole concentration range.
Okay, makes sense. So the moment we've been building up to, the results. Sensitivity first, who could detect the really faint signals better?
It was a clear win for ddPCR. Significantly higher sensitivity, especially in that crucial low concentration zone common for eDNA, say between 0.01 and 1 copy per microliter. $10 to $9,200.
Can you give us a number on that? How much better?
Let's look at the concentration needed just to get a 50-50 chance of detection, like flipping a coin. For ddPCR, across all 3 fish assays, that threshold was around 10 to 2 copies per microliter, so 0.01 copies.
Okay, very low. What about qPCR?
qPCR needed substantially more DNA to hit that same 50% detection probability. For cod and herring, it was around 10.7 copies, which is maybe 0.2 copies. But for saithe, qPCR needed a full 1 copy per microliter, $100 to get that 50% chance.
Wait, hang on. So for saithe, ddPCR could reliably detect 0.01 copies, while qPCR needed 1 copy just to have a coin flip chance. That's 100 times difference in sensitivity.
It's a massive difference. If you're looking for something rare, that could easily be the difference between finding it and thinking it's not there at all.
Absolutely. Any other striking examples?
Yeah, the saithe assay, again, at a slightly higher but still very low concentration of 10 copies per microliter, 0.1 copies. ddPCR detected it 88% of the time. qPCR, only 11% of the time.
Wow, 8 times higher probability with ddPCR at that specific point.
8 times. And this translated to the field samples, too. When they ran the fjord water samples, qPCR failed to detect DNA in 86 samples, where ddPCR got a positive hit. The ddPCR only missed in 38 samples total across the board.
So fewer false negatives with ddPCR in real-world conditions. That's compelling.
Very.
Okay, so ddPCR finds the faint signals better. What about counting them accurately? The precision part.
Finding number 2. ddPCR also showed superior quantification precision, again across all concentrations and all 3 fish assays.
How did they measure precision? Was it just less variation between replicate runs?
They used the Bayesian models to estimate the uncertainty around the concentration estimate. Think of it as the error bars, or the credible interval range. A narrower range means higher precision. You're more confident the true value is close to your measurement. qPCR consistently had a much wider range.
Meaning the qPCR results were just fuzzier.
Yeah.
Less certain.
Exactly. And the difference wasn't trivial. For the cod assay, the difference in that uncertainty range was about 1 order of magnitude, like a 10-fold difference in precision.
Okay, that's already significant.
But for herring and saithe, it was even more dramatic. Roughly 2 orders of magnitude difference in variability.
100-fold difference in precision.
Approximately, yes. So ddPCR wasn't just a little bit better at counting. In some cases, its results were arguably 100 times more precise than qPCR's for the same sample.
That feels like the real novel finding here, putting actual numbers on how much better ddPCR is, confirming what people suspected.
Precisely. They empirically measured the extent of that advantage right across the critical low concentration range.
Okay, so the study paints a strong picture favoring ddPCR, especially for tough eDNA samples. But they didn't just stop there, did they? They also looked at improving qPCR.
Right. They explored if qPCR, the older method, could be, let's say, optimized, especially where it struggles most at those low concentrations.
And they used a different mathematical approach.
Yeah. They tested a 2-step model for analyzing the qPCR data. Traditionally, you just use one model to estimate the quantity directly from the CCAT value. This approach first asks, is the target DNA even detectable above the noise, using a detection probability model? Then if it is detected, a second model estimates the quantity.
Separating detection from quantification seems logical. Did it actually help?
It did, significantly. For those samples starting with less than about 100 copies per microliter, 102 and 2, using this two-step model improved the quantification precision of qPCR by, on average, half an order of magnitude.
Half an order of magnitude. So making the results roughly 3 times more precise just by changing the math.
Something like that, yeah. A substantial improvement in reliability, specifically targeting qPCR's weak spot at low concentrations.
Does that two-step approach help when you have lots of DNA?
Interestingly, no. They found that once you got above about 10 copies per microliter, the precision of the standard qPCR model and the two-step model became pretty much the same. So really a fix for the low end.
Okay. What about pushing the absolute physical limits? Can you make either method even more sensitive if you really need to?
Yes. The paper points out that for ddPCR, the theoretical lower limit of quantification isn't fixed. You can push it lower by simply increasing the number of droplets you generate and analyze for each sample. More droplets means a better chance of capturing those rare molecules.
Makes sense. More lottery tickets, better chance of winning. What about qPCR?
For qPCR, the equivalent strategy is increasing the number of technical replicates. That means running the exact same sample multiple times in separate wells on the qPCR plate.
Does that actually lower the detection limit?
It effectively does, by reducing the statistical uncertainty. If you run it once and get a borderline high CT value, you might not trust it. But if you run it, say, 8 times and 6 of them show that same borderline signal, you gain a lot more confidence that it's real. With enough replicates, you can push qPCR's practical detection and quantification limits down to be very similar to ddPCR's.
Ah, but replicates cost time and money.
Always the trade-off.
Okay, let's pull this all together. If you're an ecologist or a lab manager planning an eDNA study after reading this, what are the big takeaways? What should change in practice?
The clearest recommendation is about choosing the right tool. The data strongly suggests that if you expect your eDNA concentrations to be below 1 copy per microliter, you really gain an advantage by using ddPCR. That's where its superior sensitivity and precision truly shine.
Below 1 copy per microliter, go digital. Got it.
But there was also a really interesting, maybe unexpected finding about ddPCR itself.
Oh, I thought the whole point was it didn't need standard curves like qPCR.
Theoretically, for basic quantification, it doesn't.
Yeah.
But the study found that the performance of the ddPCR assay could still differ depending on the specific target. Remember how the SAFE assay behaved a bit differently.
Right.
So even though you don't need standards for the final calculation, the authors strongly recommend still running standard samples when you're setting up and validating your ddPCR assay.
Why? What's the benefit then?
It helps you understand the technical performance. Is your assay amplifying efficiently? Is there something inhibiting the reaction? Running standards helps you diagnose those kinds of problems, even if you don't use the curve for the final number. It's about quality control and troubleshooting.
Okay, so standards are still useful for ddPCR, just in a different way for optimization, not quantification.
Exactly. And they also flag a little quirk. The standard software calculations for ddPCR can sometimes slightly overestimate the concentration in very low-level standard samples, something to be aware of.
Good to know. Any other universal advice that came out of this?
Yes, a really strong reinforcement of something we already knew but perhaps underappreciated. The power of technical replicates.
For both methods.
Absolutely.
Yeah.
The study hammers home that running replicates isn't just about checking your pipetting. It's a critical tool for reducing the fundamental statistical uncertainty, especially when you're working near the limit of detection. It boosts confidence in your results, whether you're using qPCR or ddPCR.
So more reps equals more reliable data, regardless of the machine.
Fundamentally, yes.
Okay, wrapping up then. This deep dive really gives us solid quantitative evidence that ddPCR generally outperforms qPCR for detecting and counting sparse eDNA targets, offering much better sensitivity and in some cases dramatically better precision. But it also shows that we shouldn't necessarily throw out qPCR, especially if cost is a major factor. Smart analysis, like that two-step model, can significantly boost its reliability at low concentrations.
And that leads to a really interesting closing thought, doesn't it? We now have clear evidence that increasing technical replicates is a powerful way to reduce uncertainty and improve detection limits for both qPCR and ddPCR.
Right.
So the big question for practical ecology becomes, where's the sweet spot?
How do you balance the upfront cost and inherent precision of ddPCR against the lower cost but potentially higher replication needs of qPCR?
Yeah.
If you have a fixed budget, do you run fewer samples on the fancy machine or more replicates on the cheaper one?
That's a tough calculation involving lab time, reagent costs, and the specific goals of the study.
Exactly. There's likely no single right answer, but this study gives us much better data to inform that trade-off between high-tech quantification and the brute force statistical power of replication. Finding that optimal balance is the next challenge.
A great point to ponder. What's the most effective way to spend those limited research funds for the most reliable ecological insights? Something for all of us in the field to think about. Thanks for joining us for the deep dive.